Thank you very much. Thank you very much to the organizers for the kind introduction and for the invitation to speak today. So I'm going to spend the next 20 minutes or so talking about an area that isn't usually talked about too much, which is the fungal aspect of the microbiome, and I'm going to focus on lung disease. So to begin, we need to appreciate what the human microbiome actually is. And really it is all the microorganisms, including their genetic material, that is within our human body. In fact, we have 10 times as many microbial than human cells, and we have 100 times more microbial genes as compared to human genes. And why do we bother looking at the microbiome, or why should we bother looking at the microbiome? Well, because we know dysbiosis, and we know this from germ-free mouse models, causes a marked alteration to human physiology and pathology. We know this, for example, from the development of abnormal cecums in germ-free mouse models, as well as the development of premature emphysema in such models. And more human studies are being conducted to illustrate to us that the microbiome needs to be understood both in health and during disease, and there's a big focus in the respiratory medicine field on dysbiosis in microbes. We're very good at characterizing microbiomes. We do this through various sequencing approaches. We're getting better at integrating other omics platforms to these data sets. But what we're really still lacking in is really strong experimental designs in a human and animal model context that can actually give us some mechanistic insight into the microbiome and its real functional value. So as a respiratory physician, unfortunately, the lung microbiome was left out of the human microbiome project when it was first instituted because lungs were thought incorrectly to be sterile, which we now know that they are not. And the work, therefore, of lung microbiome, even though this slide is slightly old, the trends remain the same in that we lag significantly behind many other microbiomes, namely the gut, oral, and skin. We have further complications when we look at lung microbiomes. First of all, at different levels of the respiratory tract, the microbes and the composition that we find are different. So sampling and where you sample and how you sample becomes really important. But also us being based in Southeast Asia, comparing our microbiomes in this continent versus that in the West is another feature that we do have to account for because our environments, our climates are very different. I do want to say that I'm going to focus the first part of this talk predominantly on the fungal part of the microbiome, the mycobiome we call it, but that there are other important aspects of the microbiome. The bacteriome has been very well studied, and the emerging application of sequencing to viromes. So to start, I'm going to pick a chronic respiratory lung disease, and my favorite one to start with is bronchiectasis. So what is bronchiectasis? It is a chronic, permanent, irreversible, and progressive dilatation of the bronchi. So we end up with these holes in the lung that you can see here on macroscopic pathology as well as on CT scan. And really a big part of the pathogenesis here is chronic infection, recurrent infection, and therefore the microbiome is of relevance. The other important point to make about this disease before we talk about some data is that this disease is very complex and that patients are, no two patients are the same. In fact, we see multiple subtypes even in a single clinic. And so we think that microbial approaches, and specifically the microbiome, may in fact help us to resolve this vast heterogeneity that exists in this disease state. And again, microbiology is an important part of this disease. A couple of years ago, a research priorities group was put together at the international level, and it was found that the microbiome, the fungal microbiome, should be a priority area for research. But before we can perform research into this, we need to understand what fungal sequencing entails, and it is a very complex field. From sample collection to preparation of the sample, needing bead-beading steps, as well as deciding what sequencing approach we would use. Once we've attained the data, mapping some of that data to establish databases in the fungal field has been particularly difficult because people tend not to whole genome sequence fungi and deposit them the way they do for bacteria. So the databases that we have that we can compare the sequences to are actually poorly populated, and this is a further limitation in being able to understand this part of the microbiome. But we do know that the microbiome is extremely important across multiple diseases, not just lung disease, but other disease states, be it chronic, which are related to infection or not. So before I talk about diseased microbiomes, we need to understand what the healthy microbiome looks like. And so to do this, we recruited 19 paired first-degree relatives locally in Singapore and found, unsurprisingly probably, that Candida and Saccharomyces were the most dominant fungi. We found a age-related relationship with Candida, but not Saccharomyces, which again was unsurprising. With this information and in collaboration with our next speaker, Niranjan, we set up this Singapore-wide bronchiectasis research platform in order to understand Asian microbiomes. But to do this, we needed to recruit patient for patient a matched patient to which we can compare that with from a different cohort. And so we partnered with Professor James Chalmers in Dundee University, who has an extensive bronchiectasis collection, probably the best in the world, and we matched each patient meticulously based on age, gender, and disease severity. So we try to remove the heterogeneity between the two patients in order to really understand if geographic features play a role and what the microbiome looks like in an Asian context. We call this study the K-Meb Study. So this is the first data that we published back in 2018 from that K-Meb study, and this is a Sankey plot, so it's red center outwards. And what you can see is from the phylum all the way out, we reach the level of genus. Now, I wish I could confidently put something in there which is species, which follows genus, but the databases are probably still too premature for us to confidently make those calls. So that's why the Sankey plot stops at genus. But what you can appreciate here is that the non-diseased microbiome is a lot more diverse as compared to the diseased microbiome in the case of bronchiectasis here. But one thing that comes to mind is the presence of Aspergillus, which is a pathogenic fungi, or potentially pathogenic fungi, which was absent in the non-diseased controls. We also performed extensive PCR in order to speciate some of this data to that level confidently, and we found some very interesting trends. All the Asian patients are marked in red dots, and the European patients in blue dots. And as you can see here, there is a marked preponderance of Aspergillus terreus in the European patients, but Aspergillus fumigatus in the Asian patients. So this session is all about clinical implications of the microbiome. Well, one really important thing that came from this study, more so for the Europeans than us, was that most of the screening for sensitization that's performed in our immunology labs at our hospital are focused on allergens that originate from Aspergillus fumigatus rather than terreus. So there's a significant number of ABPA patients, for instance, that were missed because of the assay that's been used at the immunological level at the hospital. When we look at the microbiome and compare this to various Aspergillus-associated disease states, ND being non-diseased, Aspergillus colonized, sensitized, and ABPA, what you see here is that while Aspergillus is one of a component of the microbiome, there are other fungi that do appear to be correlated with Aspergillus-associated disease, and there's a whole range of Basidiomycota fungi that we haven't even begun to understand what their role is and what they do in lung disease. But probably one of the key clinical things to come out of this was bronchiectasis, traditionally thought to be a disease of chronic infection and inflammation. But in fact, we found significant amounts of allergic sensitization to fungi in bronchiectasis, even when you match them up against allergic rhinitis from a cohort from NUS and Wang Diyun's group. Because I spoke about the heterogeneity of bronchiectasis, we tried to cluster these patients and found two distinct patient groups, one that was fungally sensitized and one that was house dust mite sensitized. The fungally sensitized group had a completely different inflammatory profile to the house dust mite sensitive group, and they were clinically worse in terms of more severe disease and poorer lung function. So really, can the microbiome help us resolve some of this heterogeneity that we see in clinical practice? So we then took these concepts and applied them to another lung disease, in this case COPD, or chronic obstructive lung disease. And we did this in both a cross-sectional as well as longitudinal fashion, recruiting patients from beyond Singapore into Malaysia, as well as partnering again with our friends in Dundee. We found significant differences in the airway microbiome in COPD. In fact, a higher diversity of fungi in COPD, which was slightly different to what we found in bronchiectasis. And secondly, but there were specific fungi that were associated with the COPD pathological state as opposed to the non-COPD or healthy state. We found again significant geographic variation to this microbiome, but interestingly, when these patients present to hospital with infective exacerbations, we found absolutely no change to their microbiome profile, be it both in diversity as well as composition. What was particularly interesting, though, is that if we did detect a loss of diversity at the point of exacerbation, this was highly predictive of mortality at 2 years, suggesting that a link between the microbiome profile and clinical outcomes in COPD could well be present. We went to look for these high-risk signatures from the microbiome and found two distinct groups. One group characterized by Saccharomyces and a lot of symptoms, but a second very high-risk group that's characterized by Aspergillus, Curvularia, and Penicillium. We found that, in fact, this particular group was associated with not just death, but increased number of exacerbations. And we know from the literature that recurrent exacerbations is linked to mortality. In order to confirm that this microbiome signature was truly present there, what we did was we obtained serum from this high-risk cluster and created specific IgE assays in collaboration with Phúc Tim Chu at NUS. And we're able to detect measurable systemic immune responses to the 3 fungi that characterize this high-risk microbiome in that high-risk cluster. Taking the bronchiectasis concept then back into COPD, we do know that in COPD we also have a highly fungally sensitized risk group. And again, this is where the microbiome comes into play. But as a respiratory physician, I'm interested in what you breathe and the air you breathe. And we know we breathe 22,000 times a day. We inhale 11,000 liters of air, and that equates to a lot of fungal spores. But when you break it down, actually, the air that we breathe is actually very poorly characterized. And while we know fungi are ubiquitously present in the environment, there was no proof of a microbial ecosystem in the atmosphere up until a few years ago. In this really excellent work performed at NTU by Stefan Schuster and his group, published in PNAS, they characterized the outdoor air microbiome in Singapore through a very robust deep sequencing approach over 5 consecutive days. And there was 2 messages that came from this particular paper. The first is that the air is more than 50% fungal, but we can only identify 3% of it, as opposed to 99% of the bacteria that were identified could be speciated. But even more interesting is that there was a circadian pattern to this air microbiome, much like we sleep at night and we wake up during the day. The microbes have very distinct patterns, and so I'm going to talk you through this. So we'll start with the top right-hand part, where you wake up early in the morning. Brown is fungi, blue is bacteria, and you can see that there's a mix of both first thing in the morning. As you progress throughout the day, 9, 11 a.m., up to the mid-afternoon, you start to see a shift towards fungi, which then are creatures of the night. And fungi predominate at night, and this cycle is repeated every day, just like we go to bed at night, we wake up in the day. Again, this is all deep sequence metagenomics. So we took this data, and I was intrigued by it, and I asked Stefan, can we cultivate the top 8 fungi? So we cultivated and confirmed the identity of the top 8 fungi that are listed there, and not all are known human pathogens. And then we took a group of our COPD patients, and together with our colleagues in NUS, developed these specific IgE assays against these fungi and found that COPD patients, there were a subgroup of them that were highly sensitized to these air fungi. This was not present in a healthy non-COPD individual. Why is that important? Because those with air sensitization or measurable air sensitization had very significant numbers of exacerbations. So this is for the first time showing that patients, particularly COPD, may be sensitized to organisms that are floating around in the air that they breathe. This has really never been thought about before. But I'm going to come back to what I said about the 3 different groups, and it's not just also about fungi, which this talk was about. Bacteria, viruses, and fungi all exist as a network And we are familiar with lots of networks, from social networks to networks of neurons to the internet being the biggest network. And so what we did in bronchiectasis was try to integrate those networks, meaning from bacteria, virus, and fungi from individual patients integrated in a holistic way using an approach called similarity network fusion. Using this approach, we found two groups of patients, one a low-risk group for exacerbations and one a high-risk group for exacerbations. Here, the bacteriomes, viromes, and microbiomes in the patients have been integrated. And in fact, we found in the high-risk patients that there were less diversity as expected, less microbial interactions, and an increased number of negative interactions, or what we would call antagonistic microbial interactions. So what happens over the course of an exacerbation, just like the COPD patients, these bronchiectasis patients present to hospital with recurrent exacerbations. And this is looking at just the composition. And if you look at bacteria, fungi, and viruses, all those pie charts look almost identical. Pre-exacerbation, during the exacerbation, and 2 weeks after antibiotics post-exacerbation. You actually see no change in the bacterium, virome, or microbiome here, which agrees with what the literature shows, and we show no differences in diversity. However, when you integrate the microbiomes and look at microbial interactions, you see a massive shift in the interactions, inter-kingdom interactions in the microbiome that's going on that is undetectable by looking at composition alone. And this somewhat settles post-antibiotics. When you look at this interactome a little more closely, you can see it has two components, what we call a core component and an ancillary component. What's particularly useful is the core component is unchanged by the exacerbation, but the ancillary component is quite dramatically shifted during the exacerbation in terms of the interactions. And most of the known pathogens that affect these patients exist in this ancillary network. So in the spirit of this session, why is this particularly clinically important? And how can we use these microbial concepts to apply them clinically? Well, in the context of bronchiectasis, we give our patients macrolide antibiotics. Macrolides have no effect on Pseudomonas, which is the predominant organism in bronchiectasis. And Pseudomonas dominant bronchiectasis patients are very widely described in the literature to have excellent clinical response to macrolides. So I put forward to you that the macrolide may not be acting on Pseudomonas, but be acting on other organisms to which Pseudomonas is interacting in order to give you that clinical benefit using the model that I've just presented. The second clinical conundrum is when we receive microbiology reports and we know an organism that's been cultured is resistant to a particular antibiotic, but many of us would have used that antibiotic clinical practice and the patient improves. And again, the interactome concept provides a potential explanation for why that may be the case, because it is targeting not necessarily that particular organism, but organisms to which that is interacting and having an indirect effect on the pathogenicity of the culprit organism. And really, the bigger question that we're trying to answer now is, do antimicrobials target interactomes rather than individual organisms? This can only be really answered in the context of placebo-controlled clinical trials with antimicrobial interventions, and we have lots of ongoing studies in order to address this. So I'm just going to stop there and just say that I hope that I've got across that the functional effects of the microbiome is really important to appreciate, that let's not forget the microbiome or the fungal aspect of the microbiome, and that microbial networks is a new concept that we do need to think about going forward. I'd like to thank my funders, the organizers for the invitation, and everybody in the team who's done a lot of this work. Thank you.
Sanjay?
Yes.
So I have a question for you.
Sure.
So in terms of the interactomes, how do you determine there's increased interaction? And this interaction, you said, is between the bacterial and the fungal, or between fungal interaction?
So interactions can be between any kingdom, within kingdoms, and between kingdoms. So how do we determine them? So this is initially a mathematical model, but in the preparation of this paper, we had to validate many of these relationships through basic microbiology. But ironically, as many of us will know, with these journals, they ask for all these experiments, and then at the end, because they believe what we're doing, and they say that the message is networks, we should take out the individual paired experiments that are not within a network. So actually, we have to validate each of those relationships in the lab. And so I think what this will tell you is which organisms are interacting, and then in that context, you can target that interaction. rather than an individual organism. So for example, if you have two organisms, one of which is very highly resistant to a range of therapies, and they have an extremely strong interaction with another organism, potentially the hypothesis here is you target the second organism and you break that connection. And by doing that, you will indirectly modify the pathogenicity of the culprit organism in which you're targeting.
Right. So this has not been tested in terms of in vitro, seeing how their growths interact with each other, or if you treat an antimicrobial, whether it changes their abundance.
So we do have some data. For example, one relationship we picked out of those networks is Aspergillus and Pseudomonas, which is a common interaction that happens in lots of lung diseases. And we do have in vitro data to confirm that. However, an experiment that's very hard to design is how that interaction in vitro plays a role in a wider network in vivo. And I don't think we've cracked what model is the best way to be able to address that. So the way to show a clinical effect is to look at interactomes across a clinical trial, for instance, that involves an antimicrobial intervention with a placebo arm, and you recreate interactomes throughout the trial to show that the interactome actually changes along with the clinical outcomes.
Thank you.
Do we have time for another question?
I think so.
We can.
Yeah.
Sure.
I was just going to ask a very interesting talk, Sanjay. To what extent do you think are the differences that you observe in the microbiome a consequence of treatment, or actually the cause of exacerbations and a cause of the progression of bronchiectasis, COPD, or other lung conditions?
Okay, so that's a great question, and a question that everybody wants to know the answer to. I don't think any of us can say confidently that we know the answer to that question, but I think pediatric studies, especially in early disease, and I think in the context of pulmonary medicine, cystic fibrosis has led the way, where they have sequenced very early CF samples, and they have shown that the microbiome is completely different to that in the adult CF patient. Now, what I think needs to happen, and I think this needs to happen across the microbiome literature, is longitudinal studies are grossly needed. And they're needed across a lifespan in order to fully understand what the role is of both therapeutic as well as environmental interactions with those microbiomes and how they evolve over time. So, great question. I'm sorry I don't have an answer, but it is an important question, especially if you're going to make clinical conclusions from it.
Yeah.
So I guess maybe we have time for one last question.
Sure.
I don't see any here, so I'll ask that. So is it good for dust mite sensitization then? Maybe we should all get dust mite sensitization instead of...
I think for bronchiectasis patients, either isn't good. So I think having bronchiectasis is not good. No, I think I wouldn't say any sensitization is good, but I would say that if you're going to choose one, I probably wouldn't choose fungal.
That's right. Okay.
All
Yeah,
right.
thank you very
Thank
much
you
for
very
your
much.
wonderful talk.